The overall goal of this methodology is to enable the characterization of the effect of extracellular matrix stiffness on bacterial infection of adherent cells in a highly quantitative manner.
This method can help answer key questions in the emerging field of host pathogen biomechanics, such as what is the role of mechanical forces in modulating bacterial infection susceptibility of host cells.
This technique helps create high-resolution time-lapse video sequences while simultaneously screening multiple conditions and automating certain procedures.
To carry out glass activation of 24 well dishes, add 500 microliters of 2 molar sodium hydroxide per 13 mm diameter well, and incubate the plates at room temperature for 1 hour. Discard the sodium hydroxide and use ultra-pure water to rinse the wells once. Then add 500 microliters of 2% 3-aminopropyl triethoxysilane in 95% ethanol to each well, and incubate them for 5 minutes. With water, rinse the wells once. Then add 500 microliters of 0.5% glutaraldehyde to each well, and incubate the plates for 30 minutes. After rinsing once with water, dry the plates at 60 degrees Celsius with the lid off. To manufacture hydrogels of tunable stiffness, prepare aqueous solutions that contain 3 to 10% of a 40% stock acrylamide solution and 0.06 to 0.6% of a 2% bisacrylamide solution, depending on the desired stiffness of the hydrogel. After this, add the water.